Hi everyone! Today I will tell you a story about silk. Silk is thinner than a human hair. It's lighter than a feather, but by weight is stronger than steel. And it's produced in ambient temperatures, using only water as a solvent in an 8 centimeter long body of a silkworm. So today we are surrounded by advanced synthetic materials, but not many of them can be compared with silk in mechanical performance, and no synthetic materials can be produced in such sustainable and minimalistic way. For example, let's take viscose, that is usually called a fake silk and used in textile industry to imitate silk. So, while having inferior mechanical properties, it is also requires harsh chemical processing with the use of concentrated acids, base, salts, and also an energy input to heat the material up to 100 degrees on different stages. So, two questions. First, why is silk so strong? And second, how can it be produced in a small worm? Have the same answer. And the answer is, of course, polymeric code, amino acid sequence of a silk proteins, which has a very specific pattern of repetitive, non-repetitive, and repetitive, and again and again and again motif. So this pattern allows, under controlled conditions inside the silk gland, under a flow and a change in a microenvironment, very gradual but constant, to fold this flexible polymer line into what we call beta sheet crystallites, extremely strong and robust crystals inside the silk. Then, these beta sheet crystals and the amorphous lines that are surrounded them are forming long nanofibrils, and nanofibrils bundle together to the structures that we already know, silk. And here is the experimental data. On your left side, you see glowing dots, which are actually the silk protein in the start of its appearance in the silk gland. It's disordered. And when we take the same protein from the exactly same animal in the later stages, just a few millimeters before it should have been extruded into the environment, we will see what we call silk nanofibrillar networks. So the nanofibrils that are already trying to interact with each other and bundle together. So let's imagine, if we could understand the material, if we could understand the principle underlying it, we could engineer a new generation of textiles that will be more robust, more strong, and more luxurious. So we will displace all the textiles that we wear close to our body with the silk. Yeah, Rehovot will blossom. Okay, but now I need to say, but. Artificial silk threads fail all over the world, not only in Israel. And the fundamental problem is that we still have a knowledge gap. We don't understand the molecular chain, and we don't know the small but very meaningful details about self-assembly in a silk gland. The empirical problem is when you take the silk outside its controlled environment, it immediately gelates, and you have no control over the material, and you cannot study it. So, let's return to the process about which I complained before, about the chemical harsh processing. So, right now, the dominant approach in silk chemistry is similar to reverse engineering. It means that we are taking the native source, silk cocoons, we cut, peel it, remove the protective shell of another glue-like protein sericin, and dissolve it with chemicals. And then we can regenerate it to fibers. But fibers still have these regenerated fibers will still have worse mechanical properties. Why? When we start from the cocoon, we have a full length, a beautiful molecular chain. But when we act on it with the chemicals and heat and pH, we damage it and disrupt. The good thing for us that these parts of a silk are still have this property to self-assembly, to fold into beta sheet crystallites. But the bad part is that self-assembly pathway is inevitably disrupted, and we cannot achieve the same result as in nature. But, and it's a happy but, because this artificial approach actually helps us to stabilize the silk in the laboratory, take the factors that either inducing, triggering the self-assembly or controlling it later, isolate them, vary them, and study them. And by this, we can reveal the design rules that nature applies to create material with such an amazing properties. And this is the purpose of my research, to destroy the cocoons and then try to build it back. And in fundamental goal, I want to have a toolkit of a nature in my hands. This is schematic of my basic experimental setup. So I'm taking dissolved silk, put it into the syringe, and pump it in a controlled manner in the specific liquid bath with a specific chemical composition, which triggers self-assembly and triggers the folding into beta sheet crystallites. And after this structural transition from liquid solution to a solid fiber, I controllably uptake it to the rotor. And that's how it works in real life. And despite its simplicity and minimalistic style, this setup actually can mimic or vary all the factors that I need, starting from chemical composition inside the solution, inside the coagulation bath, and physical parameters like a flow or a strain that is applied on the thread later. So, of course, the process was not straightforward. The first and the main challenge of my research was to make a stable solution in the lab that will mimic the self-assembly and preserve self-assembly from the native source. So here on the left you see a silk solution from a silk gland, and on the right you see my regenerated silk solution, and on both of them you can see a presence of nanofibrils. That means that in artificial conditions I can mimic a silk self-assembly and I can start to build my fiber from a building block from nanofibrils. The second obstacle was to find the exact ratio between the chemicals to induce the self-assembly and induce crystallization. So as you can see from this slide, I had a huge range in my experiments. I started from the amorphous jellyfishes, and I ended with something that looks like an industrial filament. And this is my slide. This is my favorite. It's a picture of me. Yes, so okay, it's not on this picture, but the mechanical property is still not mimicking the native silk, but the appearance of silk thread resembles the native one. So they are thin, they are continuous, and they are sometimes shiny. What is good about the artificial approach? We are no more limited by nature. So we can actually add additional functionalities to the silk. For example, I found that in my regenerated solution, I can easily blend a little bit of a fluorescent dye or conductive particles and add fluorescence or conductivity to my regenerated fibers. Moreover, let's push further. By unraveling the nature design rules for creating these materials, we can actually control the conditions. And we can actually vary the degree to which our material is crystalline. And by this, we can make not only fibrils and fibers, we can create silk-derived hydrogels, aerogels, films, coatings, nanoparticles, and even these microarchitectures. And well, when we are adding this together, the additional functionalities and the different versatile morphologies, we can use silk everywhere. That's my strong belief, that we can use it starting from textile and biomedicine, because silk is very quickly loaded with drugs and antibodies and etc. We can use it in activators and robotics, optics, soft electronics, smart sensors, and even cryptography. And that's it. Thank you very much for attention, and I hope that you learned something about silk today. And I also want to say thank you specifically for my very pro-feminist group that is consists only of boys. And my PI, Uljana Shimonovic. Thank you very much.